The overall goal of this mammalian protein expression technique is to produce milligram quantities of natively folded proteins suitable for structural, biophysical, and functional studies.
The main advantage of this technique is that it is a fast and straightforward protocol for obtaining secreted mammalian proteins at a concentration and purity required for structural studies.
Generally, we find that most issues with protein yield are due to cell health and viability. By closely monitoring cell viability and also monitoring media sugar levels, you greatly improve both protein yield and prolong the usefulness of cells. It is also very important to monitor cell density. We find that if at any time before transfection the cells exceed 2 million cells per milliliter, then protein yield is greatly reduced.
To perform large-scale culture of 293F cells, supplement 1 liter of 293F media with 10 milliliters of 100X glutamine and 5 milliliters of 100X penstrep. The antibiotic is at a sufficient strength in serum-free conditions, and the reduced antibiotic concentration improves cell viability during transfection, which improves protein yields. Culture the 293F cells in 300 milliliters of media in 1 liter polycarbonate baffled Erlenmeyer flasks with vented caps at 37 degrees Celsius with 8% carbon dioxide, while shaking in a standard tissue culture incubator. 1 day before transfection, dilute the cells to 0.5 million cells per milliliter density. On the day of transfection, supplement the culture medium by adding 10% volume of 2% weight per volume cell boost in 293.